So we're going to take a second between looking at DNA replication and the next thing, which is making proteins using the information that's in that DNA, to think about how cells change and reproduce, because that's why we make new copies of DNA. That's why DNA gets replicated, is so that when the cell splits into two cells, each copy of the parent cell has enough DNA. If we don't split the DNA, one of those daughter cells isn't going to have any DNA, isn't going to be able to produce any proteins, isn't going to make any daughter cells of its own, and that's not good. All right, so just like what you might have heard of, like life cycles, all right, this is the cell cycle. The cell cycle is the process that they go through as they kind of grow up, all right? So we're going to start right here on this part of the circle as what's called mitosis has just happened, all right? So mitosis occurs, and we've got a brand new cell, all right, with DNA that's 50% old, 50% new, right, semi-conserved. It's in the G1 phase, right? It's going to either grow and reproduce, go around the cell cycle, or it's going to grow and die. That dying is, if it's on purpose, it's called apoptosis. That's programmed cell death. The lysosomes break apart, and all the chemicals that were inside digesting and breaking down things start digesting and breaking down everything in the cell, right? But what can also happen is this G0 phase, sometimes called G0, N-A-U-T, all right? And that just means that it's not really growing, it's not really developing, it's just at where it's going to be, all right? This is kind of rare. Most cells are going through the cell cycle of growing and reproducing or dying, right? Very rarely, overall, like all the cells in the world, does this happen, all right? So this is going to be making exact copies, right? We want clones, no mutations, no differences, because your skin cells want to make skin cells, right? The cells on the lining of your stomach want to make more cells for the lining of your stomach. These are the steps, but don't write them down because we're going to talk through each of them. All right, so interphase, I have this as a little asterisk because it's kind of part of mitosis and it's kind of not. All right, so it's not really part of mitosis, it's what's happening outside of mitosis. All right, so we've got the in the G1 phase, the cytoplasm is going to double and all the organelles are going to double. So the cell is going to make all of that stuff. S phase is DNA replication, so what we just talked about last section. G2 is error checking, so it makes sure everything's the way it should be before mitosis happens. Because if it's not the way it should be before mitosis happens, well, you just copied a mistake. And now you got two mistakes instead of just one. Prophase, you might remember pro as in before, right, is the first real step of mitosis. And we've got those chromosomes, those coiled up bundles of DNA. They are going to move into this like fully coiled up, and we're going to get this kind of classic X view of a lot of the chromosomes, all right, where the DNA is replicated and those two replications are going to be joined by something called a centromere, all right, a centromere, and it's going to have two sister chromatids. Those are the strands of DNA here. I know it's kind of just a vocab thing. So it's the replicated DNA. Sister chromatid and sister chromatid are joined together. All right. And then what we've got is the nuclear envelope disintegrating, so no more nucleus. All right. It's still a eukaryote, but there's no nucleus for a little bit. All right. Metaphase is going to look very much like this, where the chromosomes that we just talked about are going to be lined up in the middle. Think middle, all right? And they actually line up on something called the metaphase plate. It's not a real thing, it's more like an imaginary line, all right? But they line up because all of those centromeres are pulled by something called centrosomes, All right, so the centrosomes are at the poles, all right, and they are pulling the centromeres so that everything's lined up in the center. And it's lined up in the center because during anaphase, they're going to get pulled apart. So those spindle fibers are pulling with such force that the chromosomes are physically pulled apart. All right, so sister chromatids are being broken down as enzymes are breaking them apart, and the spindle fibers are pulling them to opposite sides of the cell. And we've got another vocab change. Sister chromatids are now called daughter chromosomes. You heard me call the new molecules of DNA daughter molecules. All right, so this is that daughter chromosome. All right, same concept here. The daughter chromosomes are going to be pulled towards the opposite ends of the cell, and I got a picture for telophase too. All right, but telophase is kicked off once those chromosomes get to the poles of the cell. And as that occurs, nuclei are going to come back. So we're going to have nuclear membranes reforming. Chromosomes are going to start to uncoil. All right, those mitotic spindles are going to go back away, and a process called cytokinesis starts. Telophase is kind of the opposite of prophase. All right, cytokinesis looks a little different if you're a plant or a fungus versus an animal because of the cell walls. All right, cell walls are going to start with vesicles lining up in the center, pretty much where that metaphase plate was. All right, and the vesicles are going to fuse together. They're actually going to join together and form the cell plate. The cell plate has cellulose added to it. If you're in a plant, keratin added to it if you're in a fungi, and a cell wall forms there. Animal cells, though, it's going to be pinched together like you're pinching a balloon, right? And it's going to divide all the cytoplasm and the organelles in half, 50-50. All right, if you're a visual learner like I am, this is all of that information, but in a diagram with some pictures. All right, so end of G2 phase, error checking is done. We've got our centromeres. We've got all the DNA, twice as much as a regular cell would have. Prophase, the nuclear envelope starts to go away. The DNA coils into chromosomes, and the centromeres are going to move, or the centrosomes, my apologies, is going to move to the poles, like we see here. All right. This diagram has something called prometaphase. You do not need to worry about that if you are in my regular biology class. Honors biology, you do. All right. So honors biology, real quick. This is kind of a 50-50 split between what we see in prophase and what we see in metaphase. All right. We have the nuclear envelope continuing to go away. We have microtubules connecting to the centromeres, all right, which I know this one labels it as a kinetochore. We're just going to keep calling it a centromere. All right. And back to everybody now. All right. So biology and honors biology both. Metaphase, remember, it lines up in the middle. So all those chromosomes are going to be pulled to the middle by the spindle fibers pulling on their centrosomes. Enzymes are going to then break those centrosomes down. The spindle fibers are going to shorten, and it's going to pull the chromosomes apart into daughter chromosomes. Then we've got a cleavage furrow happening because this is an animal cell. All right, so that contractile ring is squeezing, and it's going to pinch together, making two new cells. We're going to call those daughter cells. All right, here's just another way to view this, trying to give you guys quite a few in your readings. There were also a couple different diagrams as well. All right, so as we're thinking about this, this is mitosis. The other way that we grow and reproduce is something called meiosis, but we're not going to talk about that right now. We'll talk about that later when we talk more about genetics. All right, so everybody needs to know prophase, metaphase, anaphase, telophase. They need to know the cell cycle. They need to know interphase. They need to know what the process of cytokinesis is. Honors, you also need to know prometaphase. Right? I always use the acronym that I learned as a mnemonic I party more at the club. So if you write those first letters, you get I, P, M, A, T, C for prophase, metaphase, anaphase, telophase, cytokinesis. There are quite a few out there if that one doesn't work for you, but it works for me. So remember, have all this in your notes. For today, though, instead of asking a couple questions, I want you to either draw this or this in your notes. Just some easy diagrams. I think this one's probably easier to draw. I think this one may be more helpful to some of you, especially my honors class who has this part as well. And I'll check your notes tomorrow.